Hi, everyone. Before we start, I quickly wanted to introduce ourselves so that everyone knows who we are. I'm Sabine Wintergerst. I started actually as a Fairchild volunteer and then upgraded to Seed Lab Manager in 2019. And since then, I'm working there. And next to me is Brian.
I'm Brian Harding. I'm the horticulturist for conservation. I've been here since 2018? Yeah. 2018. Yeah. Wow. So.
Okay. So thank you for tuning in to our little field trip through the Seed Lab and Fairchild's native plant nursery. And yeah, so we're gonna first start with the Seed Lab, and then I'll hand it over to Brian. He's gonna show you all the plants. So before we enter the Seed Lab, actually, I wanted to show you some of our really beautiful pine Rockland seeds. And like the plants themselves, they really come in all kinds of colors and shapes. And all these images were taken by Jack Hahn, our Fairchild volunteer in the imaging lab. And for me, these pictures are just really putting a spotlight on seeds that are often overlooked, even though they're so important for plants, not only for reproduction, but when you think about it, they're also the only way plants can move from one place to the other. And also just to see all these details on the seeds that you normally don't see without a magnifying glass, it's really amazing. So, but now what actually is a seed bank, or what's the role of a seed bank? So the seed bank is basically provides a safety net against the extinction in the wild, especially for rare, endemic, threatened, and endemic species. And it's basically, we store, especially species that have a narrow range, they're really threatened by catastrophic events like hurricanes. And here in Florida, we have these species that only occur in a few islands of the Keys. And so it's really important to safeguard their genetic diversity in case of an emergency. And we can use these seeds to regrow the plants. But we also use the seeds to grow plants for our Connect to Protect network, for example. And we also just grow plants from these seeds that we store for all our projects, the reintroductions, the augmentations. And the seeds also get used for seed research, because a lot of the native plants, we actually don't know that much about how to germinate them, if they can actually be stored or not. And also seeds can be used directly for introductions or reintroductions. But what happens to a seed when it enters the seed lab? So the first part is the seed cleaning, which is the most tedious and often most time-consuming part. And I'm grateful for all volunteers and students and interns that help with that. And here you can see two biotech high school students that helped last year, Melissa Silva and Janae Bailey. And they are getting blotched silverbush seeds out of these organza bags that we use to collect them. But often seeds come with a lot of chaff and flower parts. And here you see seeds collected from Florida white top seeds that were collected by Patty, which I think is on the call, on this Zoom call as well. And thank you for that, Patty. And so these seeds are really tiny black seeds. I think this is a seed. And so cleaning them from all this chaff by hand would be really, really time consuming. And so that's where my most favorite tool in the seed lab comes into play, which is the seed blower that you can see here. It's this machine that is basically a pump that pumps air from the bottom to the top. And you put the seeds and the chaff here on the bottom. And then when you start it, you can control the airflow. And normally the chaff is lighter than the seeds. And so the chaff starts flying up and gets caught here in these little barriers, whereas the seeds stay on the bottom. And sometimes you have to repeat it a few times, but at the end you get clean seeds without spending hours doing that by hand. Oops, how do I do that? And then before the seeds are actually stored in the freezer, we also assess the seed viability, which is really important because for a lot of seeds, we don't know how long they will store in this freezer. And so we want to have a baseline germination that we can compare the germination rate of seeds that we retest after several years. And also by testing our collections every 1 to 5 years, we also get an idea when the collection actually starts to lose viability. And that's then a sign that we have to use the seeds and recollect the seeds. And we do these germination trials in petri dishes. And here you see Keith's partridge pea seeds germinating on filter paper. And here we also use agar to germinate the seeds. Here you have the mistletoe cactus seeds. And we do these germination trials in the incubation chamber, which allows us to control humidity, temperature, and light. And that makes sure that we can repeat the experiment under the exactly same conditions years later. And then before the seeds go into the freezer, they have to be desiccated because seeds are living cells and they contain a lot of water. And as you all know, once you put water in the freezer, it freezes and it expands, and that can damage the cells. And normally we use a salt to dry the seeds. Here we use magnesium chloride, which has the advantage of bringing the relative humidity in the desiccator to a really constant level. And once the seeds are dried, they get sealed into these mylar bags and put into the freezer at -18 degrees. We also do a lot of seed research. And like I said before, for these native plants, we don't really know sometimes if they actually survive the freezing. And here in Florida, especially, we have this unique situation that we have tropical and temperate species here. And tropical species are the ones that tend to, there are more tropical species that don't survive freezing than in the temperate zone. And here is one example. It's the Kunti, which you all know. And here are the huge amount of Kunti seeds that were cleaned by Joanne, also a volunteer who's really, really good at that because they're actually hard to clean. But Kunti is one of these species that you can not store. So they survive desiccation, but they die in the freezer. And another part of the seed research is to actually find methods to break the seed dormancy, because a lot of seeds, they, when you put them into the soil, they're not necessarily germinating right away. So in nature, they're basically waiting for the right conditions to germinate. And since we are working a lot with pine Rockland species, fire might be one of these cues that actually triggers germination in a lot of species. Like most of you, or I think a lot of you have been in a pine Rockland after a burn and you see all these bulbs resprouting. And so it might be that the particles from the smoke actually trigger the seed germination. And to get the fire into the seed lab, basically, we produced a so-called smoke solution by burning pine straw in this bee smoker and then guiding the smoke directly through the heater hose into this sidearm flask that was filled with water a little bit. And then the smoke had to basically bubble through the water, and all the water-soluble chemicals of the smoke get trapped in the water, and that's what we can use to treat the seeds. And the biggest success we had with blotched silverbush, which is an endangered species. And I think Jen mentioned it in her talk before, but I just wanted to show it again because it was so impressive that at the same time without smoke, there was maybe one seed germinating right here. But when you look at the seeds that were treated with smoke, you have like, yeah, most of them came up, and it's really nice. And another thing that we often do, which is kind of an intersection of Brian and me, is the seed bulking, because sometimes we don't have enough wild collected seeds, and we also don't want to over collect wild populations and negatively impact them. And so what we do is we collect some wild seeds and then grow the plants in our nursery and then collect the seeds from them. Here you see the Keith's partridge pea, where we also use these organza bags to bag the seed pod so that we can actually collect the mature seeds when they get ejected. And in this case, for example, we use these huge amount of seeds that we got from that to do a little study about how increasing soil salinity affects seed germination and seedling survival. And with that, I give over to Brian.
We're gonna swap around a minute here. Oh, where are these masks? Okay. So Sabine and I definitely work in tandem. So a lot of the research that she does has direct implications on what we are able to grow at the nursery. So this is a typical scene at the nursery. We work with about 50 or more than 50 different pine Rockland species. So one thing that we want to do, the kind of the goal in this Connect to Protect, is to incorporate these plants found in the wild, back into Miami-Dade or the Monroe Keys urban matrix. And so we'll use Mule Grass here as an example. So this is Muley found on Big Pine Key in full bloom. And so I rely on my wonderful volunteers to help me collect. And I am so sorry I didn't have a picture of the volunteers collecting Muley, but this is them collecting Schizochyrium gracili, the wire blue stem at Martinez. But Muley's collected in the same fashion. And so this is Kim Bavuzo, Isabel Lamas, and Sharon Ladner here at Martinez. But the goal is to reproduce these plants in large enough blocks to be able to fuel the program, to fuel Connect to Protect. And as you can see over here on the left, this is our large block of Mule Grass. These were collected at 2 different pinelands, at Ned Glen and at the Deering Estate. And then over here on the right, you see the final product. This is a superlative planting over along Old Cutler Road in Cutler Bay. And you can see how Muley makes an excellent addition here in the urban landscape. So part of the goal in this is to be able to offer plants that you should not be able to walk into a typical nursery and find. And we reproduce them in 2 different ways. On the left, you'll see reproduction by seeds. So I've got Angadenia bertoii, or the Pineland allamanda, and Pityopsis graminifolia, or the narrowleaf silkgrass. So these seeds are just getting their start here. And over on the right, this is reproduction by cuttings. So these cuttings, this is Choanophyllum villosum, or the shrub thoroughwort. So this one had an interesting story. We actually collected these at a Wasserbauer edition. So these were plants that were occurring in the footprint of a road widening project along Chrome Avenue. So what we were able to do is to collect at least 2 cuttings per plant. And so we did not rescue the original plants, but theoretically reproduce them through this process, and then sent them out as fully rooted plants through Connect to Protect. So one thing that I like to do is I like to see a plant in the wild, because a lot of times that informs what I do at the nursery. So in this case, you're looking at a plant called Bahama Saxia. And as you can see here on the left, it's rooted into the Miami oolite. If I attempted to grow this in moisture retentive potting soil that's heavily fortified with peat, this plant would not be happy. So in this case, it actually informed my decision to put this in a mix that a lot of our plants respond well to. So this is only 1/3 potting soil. It's 1/3 crushed limestone and 1/3 turfus. And this turfus, think of that as a finely chipped up clay pot. It holds just enough moisture without holding excessive moisture. So I like that because it's fortified with calcium clays. And since our pine Rockland species occur on limestone, this has a good cation exchange capacity for the time release nutrients that we use to fortify the soil. None of this would be possible without our wonderful team of volunteers. Over on the left, you see just a small portion. I probably have 15 to 20 people who volunteer their time 4 days a week. And they're usually with me anywhere from 3 hours to Mary Jackson over here on the right is usually with me the entire day. They are so instrumental in this process, from everything from, as Sabine mentioned, seed cleaning, seed collecting, sowing, advancing material from, community pots to 3-inch to gallons, and then they help, distribute the plants through the Connect to Protect distribution. So this is one of my little pet projects. Some of the plants that we work with need a little extra attention. This is one. So you're looking at Milanthro Pervifolia, or the Pineland black anthers. So over on the left, you see a plant at the Deering estate. And over here on the right, you're going to see multiple pots with up to 5 cuttings per pot. We've been able to collect seeds of this in the past, but we've not been able to germinate them well. We've had practically no germination. So one of the goals of this was to be able to, each of these pots over here on the right represent a single plant, 5 cuttings per plant, and they're from different pinelands. And so in this way, we've been able to capture a small genetic sampling from all of the different pinelands, and without necessarily impacting, the plants in the wild. We root them, grow them out, and so the image you see here in the middle is a reproduction of a wild collected plant by cuttings. And so the goal is going to be to get all of these different genetic lines together in one place, and with the goal of improving seed set, because we think this needs to be outcrossed. We'll get better seed that way. And so this is just a typical scene from the nursery. And on this note, I kind of have to thank everyone, because this is a team effort. We rely heavily on our field biologists, Jimmy Lang and Lydia Cooney, to collect seeds in the wild, Sabine to do the seed research, Daniela Champney, I really need to thank her. She is the manager of Connect to Protect. She is responsible for getting the material from this point out to the general public. And all of this is overseen by Jennifer Posley. We really need to thank all of our volunteers, all of our interns, the Miami-Dade County EEL program and the Parks Department for allowing access into the preserves. And we want to thank all of our partners. So, Sabine?
Yay! I just can second that. So I have nothing to add to that. But thank you to everyone who helps us making this possible.
Take a couple of questions from the audience.
Can we, is there any questions that anyone has?
I'm telling people to unmute themselves or just put it in the chat. So the potting medium is, could you talk a little bit, Brian, about Turfus?
So Turfus was a product that I was not familiar with prior to moving here. And I believe they use it to repair baseball diamonds. And from what I understand, there's 3 different sizes of the Turfus. We only use the middle and the large. And it's a little bit more, because it's fortified with calcium clays, it's a little bit more on the basic side, which is perfect for all of our pine rockland species, since they are occurring on limestone. That middle grade is fantastic for seed germination. The larger grade, we actually will incorporate that into the mix. And so there are some plants, the Bahama Saxia, the Brackelia,
Asclepias.
Yeah, the Asclepias, the butterfly weeds that respond really well because they don't want moisture being held right at the root system. So this would only hold a limited amount of moisture and then, quickly releases the rest of it. Does that answer your question?
I bet it does. We have a lot of accolades for you guys coming in. Here's a question that's it's for both of you, but from John Kalifarski, he says, have you had to scarify seeds to get them to germinate?
That's
Yes.
And maybe repeat the question.
I forget that you can't hear Jen. Jen is reading us the question. So, John was asked.
John says, have you had to scarify seeds to get them to germinate? Do you think it needs to pass through an animal's gut?
So the question was if we had to scarify seeds to germinate them, and if some seeds have to pass through an animal's gut to germinate. So both, so we scarify a lot of seeds. So that's the first thing. And a lot of seeds react really well to it. And for me, I do it to grow plants. I always would like to know what actually triggers germination in nature, because scarification with a knife or sandpaper, it's kind of artificial, but it does help to germinate a lot of seeds, especially seeds of the bean family. Most of them need scarification to germinate well. The other thing about the seeds passing through a gut, yes, I think some definitely do. There are some seeds that really, I think it improves germination of some species, and we do have some seeds that we treat with sulfuric acid to basically mimic the acid that the seeds will experience in the gut of an animal. And yeah, so for example, it's not a pine Rockland species, but what comes to my mind is the key tree cactus. And you have the cactus fruits and they get eaten by animals. And when you scarify the seeds with sulfuric acids, they germinate much better. So that's an example for that.
Here's a question for both of you. Brian, what's your biggest propagation breakthrough that you would like to talk about?
Oh my gosh.
I can't think of one, maybe Sabine. I bet you both can.
Well, I need to think about this in a minute. My biggest propagation breakthrough. I'm looking at you too.
You can think about it, and I can ask you the next question.
Yeah, well, I'm going to think about this one.
Okay. Okay, here's a question for both of you from MD Sanchez, and the question just rolled away.
Okay.
I'm using this through the web, and the interface is really clunky. But it said something about, do you ever do controlled pollinations in order to generate more seed?
And we haven't done it so far, but actually with our Asclepias, the butterfly weed project, we're actually thinking about it. So we have a few plants at the nursery that are mature now. Especially from what's the common name of the Asclepias? Where it is?
The green antelope.
The green antelope horn. So that Brian grew like 16, 15 plants, and they are flowering now for several months, and they never got pollinated so far. So we're definitely thinking about trying hand pollination for the butterfly weeds, but we haven't done it. Or we tried it once, but it hasn't worked, and we're gonna try again and see if we can get some seeds from them.
Here's a question. Do you have any seeds that have a really delicate embryo and need special handling? That's from John.
A delicate embryo. No, not really. Basically, not yet. Yeah, that I can say. I don't work with orchid seeds. I don't work with fern spores, which are not really seeds, but go into this category. Most of our seeds have really hard seed coats, and it's actually hard to get them to germinate and come out of their seed coat. So I guess, yeah. Do we have to think?
Well, I just want to go back to kind of the difficult to really, when the plants are up, I really don't have much in the way of issues growing some things. I think the biggest challenge has been, collecting stuff, and it's really in the breaking the seed dormancy. So, and this is where, Sabine doing, the research to break the dormancy on many of these things has come into play. So I would say, that to me is more the big breakthrough. Once the plants are up, the growing conditions are, they're not the same for everything. I think for me, it's mostly finding out, if they want to be reproduced in the sun or reproduced under 40% shade, or if the seedlings want to be kept, drier when they're smaller. It's just little things along that line for me.
Here's a question from Naki. He says, do you ever incorporate local soil or other material for ectomycorrhizal inoculation? In the Turks and Caicos Islands, we have found it crucial for several species, including pines and coca loba.
So from there, I do use a product that has some mycorrhiza in it. It's a composted mix. It's got bat guano and earthworm casting along with the mycorrhiza. So, and we have found that that's been really nice to add to the mix on several of these plants. They've responded very well to that.
We got two for Sabine. Nick would like to know, do you use tissue culture propagation?
Someone wanted to know if we use tissue culture propagation, and we don't so far. So we only do normal germination trials with the seeds, not tissue.
And then Naki said, can you confirm the acid you used for the seed, for the cactus seed, was it salicylic acid?
It was sulfuric acid. And we used it. So, sorry, I have to repeat the question. Sorry. Naki asked if to specify the acid we used for the key tree cactus. So we used sulfuric acid, and we used it in 5% concentration. And the seeds were soaked in there for 5 minutes and then rinsed for 15 minutes in water.
That's coming to the end. Do either of you want to talk about a biggest propagation breakthrough?
The biggest propagation breakthrough for me was definitely the blotched silver bush that I showed before, because it was the first time I used the smoke solution, and it was the first species that tried it on, and it was just a crazy difference, and that was really amazing for me to see. I think that, yeah.
Yeah, my biggest one, like I said, I think is just the seed breakthroughs, which is where Sabine has been like a fantastic addition to this program, because she's really been able to increase the number of plants that we work with, because breaking that seed dormancy was such an issue on some of these species.
All right.
And they go over to you.
Steve says he doesn't know about us. We seem kind of seedy. That's so. I think if there are no more questions.
So I think Jen is through with the questions.
Yeah.
Come, Jen.
Hi and bye.
So we're gonna say bye.
We'll kill the meeting. Thank you. Thank you for joining, everybody.
Thank you.
Thank you.
I don't remember how to end the meeting. No.
Maybe you have to do it on you.
Yeah.